disconnect at this time. I would now like to turn the meeting over to Irene Ihier. You may begin.
Thank you. Hello, I am Irene Ihier of CDRH's Office of Communication and Education. Welcome to the FDA's 33rd in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality here in CDRH, will provide a brief update. She is joined by Dr. Brittany Shook and Dr. Chris Roth. Following opening remarks, we will open the line for your questions related to today's discussion. Please remember that we are not able to respond to questions about specific submissions that might be under review. Now, I give you Toby.
Thanks, Irene, and thanks everyone for joining us again this week. So I just have a quick update. You may have seen that yesterday we issued a letter to clinical laboratory staff and healthcare providers regarding false positive results with antigen tests. So we put that out because we have become aware of reports of false positive results in nursing homes and other settings. And as we continue to monitor and evaluate these reports, we wanted to share some information with stakeholders about this risk and about steps that can be taken by clinical laboratory staff and healthcare providers to help ensure the accurate reporting of test results. So that's available on our website and was also sent out by email yesterday. So a few of the key points there are to be aware of the information in the package insert for each test, which does include information about appropriate handling of the test cartridge or card, excuse me, and appropriate storage of those components. If they're not handled properly, that can impact the performance of the test. And also about the time that it takes to read the results and being sure to read the results at the appropriate time, since reading the test before or after that time frame can result in false positive or false negative results. We also include some information about minimizing the risk of cross-contamination and pointing to some CDC guidance and recommendations about the use of PPE and changing gloves, and also cleaning the instrument and the work area in between specimens. Then the other point that is made in that communication is about the positive predictive value, and just reminding everyone that false positives are expected with all tests. You know, any test that's out there will have some false positives and some false negatives. No test is perfect, and particularly with some of these tests being used to screen individuals in low prevalence areas, we do expect there to be a higher incidence of false positives as that prevalence decreases. So you can find that on our website and take a look at that and let us know if you have any questions. The only other update I have is just to remind everyone that we will not have a town hall next week. Wednesday of next week is Veterans Day, so we will be taking a break next week and we'll be back on the 18th. So with that, we can open the line for questions.
If you would like to ask a question, please press *1 and record your name clearly when prompted. To withdraw your question, you may press *2. One moment, please, for our first question. Our first question comes from Mark Hackman. Your line is open.
Yes, good morning. Can you hear me?
Yes, good morning, or good afternoon, I guess.
Yes, good afternoon. I'm sorry, it's morning on the West Coast still. Morning, Toby. It's Mark Hackman. It's been a while since I've spoken with you. It's been a couple months since I've been on this call, but I'm going to ask the same question that I asked a couple months ago, and it's kind of a two-part question, but they're really short. My first part is, when are we going to see a fully at-home test approved by FDA? And my second part of the question is there currently any over-the-counter devices, and I'm talking about lateral flow devices, that have been approved by FDA to this date? I've checked the website and I didn't see any.
Thanks for joining us. I'm glad you've come back. I'll start with the second half of your question. There are currently no over-the-counter COVID-19 tests authorized. We are working with any interested parties that come to us with, looking to do that, and we have put out some information in our templates about over-the-counter. So we do look forward to that. And the same is true for fully at-home tests. I can't give you any timeline on that, unfortunately, because that is dependent on developers submitting those EUA requests and making sure that they are that the data is there to support that use. But we have, as we've talked about on this call previously, we are very interested in that, and we have put out information about, our recommendations as well as our flexibility to consider different approaches. So we are looking forward to getting to that point.
Thank you very much. Have a great day.
You too.
Our next question comes from Shannon Clark. Your line is open.
Hello, this is Shannon Clark from UserWise Consulting, a human factors usability testing consultancy. I'd like to better understand the FDA's rationale for differences between the molecular and antigen templates, specifically related to studies to support a point of care claim. So not OTC or home use, but point of care. There are pretty significant differences between the molecular and antigen templates, where the molecular template specifies at least 4 different operators, while the antigen template remains silent on the matter. The molecular template specifies prospectively collected positive specimens, while the antigen template allows for either prospectively or retrospectively collected samples. And also, the molecular template implies no training for healthcare professionals, which is very common for human factors and usability testing, while the antigen template does not comment regarding the use of instructions. So if you're allowing retrospective sample collection for the antigen template, but not for the other one, as well as just one healthcare provider instead of 4, does this imply that the FDA is uninterested in the process for collecting the sample when using antigen tests in the point of care setting? And then if so, can we use just one extensively trained healthcare provider for the antigen point of care study to support a point of care indication?
There's a lot in that question. I'm going to try to try to get to it, but so there are differences. Obviously there are differences in the templates because they are for different types of tests, and that's why there are different templates for them. I would have to double check the details. I unfortunately don't have all the templates completely memorized, especially as we update them. But I believe that we do request multiple operators for the different templates, and I believe that there's the differences in the test design. Visually read versus a more complex instrument would also impact our recommendations for the study design.
Also, perhaps molecular solutions are more complex and require more operators, maybe more evidence that untrained users can use it, while antigen tests might be so simple that maybe you're less interested in having more than one operator, et cetera.
Right, and as I said, they are very different types of tests, with most antigen tests being visually read and molecular tests having additional steps involved.
It just seems surprising to me that both. What was that?
Oh, sorry. Go ahead, Shannon. I was also going to ask Chris if he wanted to weigh in at all on this question.
Oh yes, please, Chris.
Yeah, thanks. No, it is a good question, and we do struggle. I would say struggle, but we do try to accommodate, different approaches for these two different tests, right? So we know antigen is slightly less sensitive than maybe many of the molecular tests, and therefore the testing can be slightly different. And again, I think the first part of your question, which may be of most interest, is I think molecular tests have instrumentation, and you can have a visually read antigen test, and so those need to be kind of taken into account when the validation is being performed.
Yeah, so I guess when I read between the lines of these templates, it just seems that the FDA is uninterested in the sample collection, because many times both antigen and molecular will employ a swab. And I know when we do sample collection studies for home use, we're very interested in users' ability to correctly use that swab. But the implication here to me is that for point of care use, you're not that interested in swab, as you do allow prospectively collected samples, as well as not requiring.
I don't think that was the intended implication, and it would be very helpful if there are things that you're reading in the templates that you think are confusing, or you think are discrepancies between the templates that don't appear to make sense to you. If you can send those in to the EUA mailbox, that would be very helpful, so that we can take a look at that and make sure that there is no unintentional discrepancies.
Excellent, will do. Thank you so much.
Great.
Our next question comes from Christy Bergerson. Your line is open.
Hi, Toby, Chris, and Brittany. This is Christy Bergerson from Exponent Consulting. My question is not as detailed. I had a question pertaining to research use only components that are used as part of a submission. So after the submission is authorized, do those components named in that submission, which might have been research use only before, need to change their labeling? So for example, if a company uses a certain instrument with their assay and that instrument is labeled as RUO, after authorization, would that instrument marketing materials need to be changed to remove the RUO label?
So if you're distributing instrumentation that is covered under your EUA, it should be labeled as an EUA instrument, yes.
Okay. Okay, thank you.
Great.
Our next question comes from Tom McDougall. Your line is open.
Hi, Toby. Hi, all. I had a quick question about the study to support the asymptomatic claim that's in the new antigen template. I see it's now asking for 20 asymptomatic positives, but also mentions that it may be possible to use archived specimens. And I just wanted to ask if you guys could maybe shed some light on what any maybe post-authorization requirements would be if we were to go forward with archived specimens.
Oh, that's an interesting question. Chris, do you know if we're including post-authorization studies or requirements if they use archived specimens?
So, I mean, typically we'll have a post-authorization study if there's some kind of open questions at the end of your submission. So if there are, I think during the review, this is a very detailed question, right? I think there's a lot of aspects to what go into a post-market study or not. And so I would tend to leave that up to the individual reviewer, and I would say there's not kind of a blanket policy. So, yes, if you use archive, you must have a post-market study. I don't think that is the approach. I think there are maybe some deficiencies that could be identified because we know that the archive samples are, or can be at least somewhat of a biased population. So if there are those questions at the end of the study, then, that may be applicable for a kind of a post-market approach or post-authorization approach, excuse me.
Yeah, I think that would be a good question to discuss with your review team. I did just pull up the template to take a look, and the discussion about archived specimens does ask that you contact FDA to discuss those alternate study designs. So that would be helpful for this one.
Great, thank you so much.
Sure.
Our next question comes from Mark Wagner. Your line is open.
Hi, thank you. I was wondering if you could comment on what the FDA is looking for from distributors of at-home collection tests. Are there any requirements for distributors of an EUA authorized at-home collection test when the distributor does not hold the EUA?
Yeah, so the all of the EUAs include conditions of authorization for distributors in the authorization. So those would be spelled out there. And if you have any specific questions about those, that would be a good question to send in to the mailbox so that we can make sure that we understand the specific situation and can provide the appropriate feedback.
Okay, thank you.
Sure.
Our next question comes from Todd Lewis. Your line is open.
Hello, my name is Todd Lewis from DNA Diagnostics Center. And my question is, if we have a EUA-approved say molecular test, and we wanted to change the sample collection part of that, and there was an EUA-approved, say it was approved for nasal swabs, and we wanted to use saliva collection that was approved, how should we go about that? Do we have to send in a brand new EUA, or is there some type of bridging study, or we do a verification study?
So so we do have recommendations in the molecular template for validation of saliva, including validating adding saliva as a specimen to a test that has been already validated for nasal specimens. In terms of the regulatory approach, is this your own EUA that you are the EUA holder and you're looking to add saliva, or are you a laboratory that is looking to add saliva testing to a commercial test kit?
We were going to use somebody else's EUA, but we can do it as an LDT too. I mean, we could flip it around that way. But I didn't know if you could marry the two and just do a verification, and I guess that'd be LDT. Yeah.
So so if you're looking to add saliva as a specimen type to an EUA authorization, we would just ask that you submit, the EUA holder should submit a supplemental EUA request, including the validation that we recommend in the template for saliva, and then that could be added, provided that the data supports it, that could be added to the authorization. For a lab looking to add saliva as a specimen type to another entity's authorized test, we would also hope that you would use the FDA recommendations for validation that are in the template. We have indicated in our guidance, and there's also the HHS LDT statement that we would not expect to see an EUA request for that modification, but we would expect it to be done for the saliva collection to be done with a legally marketed saliva collection device. The collection device otherwise would need to come in for authorization if it's not one that is already authorized.
Right. Okay. And it doesn't have to be standalone. It could be part of another test kit.
Can you clarify that?
So some test kits have that in the collection device built into that EUA, and some collection devices have an EUA just a standalone.
Right. So if there's a collection device that is included in within an EUA for an assay, then that collection device is only authorized for use with that assay. In order for a collection device to be more broadly legally marketed, it has to have that standalone authorization.
Okay. All right. That answers it. Thank you.
Great.
Our next question comes from Roberta Bastion. Your line is open.
Hi, yes, my name is Roberta Bastion. I'm from Tempest Labs. Just a question about if you have a test authorized under an EUA and you do receive a false positive, are you required to report that via the MDR process?
We would expect false positives to be reported both to the EUA holder. It sounds like you are the EUA holder, and to FDA. So you could send that into the through the MDR process or through the EUA reporting mailbox that should be on your letter of authorization.
Thank you very much.
Sure.
Our next question comes from Elaine Allen. Your line is open.
Hi, this is Elaine Allen. I'm calling from Agilent. I'd like to first appeal to everyone and all the listeners for their patience, because today is my second day in the environment of developing tests for the COVID. So my questions are very basic. May I ask, and I have a few, what is the average approval time from submission for an EUA to final approval?
I don't believe we have averages that we've been giving out at the moment. We do have some information on our FAQs that talks about the time for review, and generally it does vary based on the quality of the submission as well as where the submission falls in our priorities.
Okay, that's great. And does the approval time perhaps, is it affected if it's a serology testing or antigen testing type EUA?
We do have different teams that are working on serology, antigen, and molecular tests. So they are each they each have their own queues of tests that they're working through.
Okay, so it depends on them. Now, the minimum test requirements is 30 positive and 30 negative for clinical data. Is that still the acceptable sample size for EUA approval?
So the recommendations for validation are in the templates on our website, and those are all of the current recommendations are in there.
Look at templates. Okay, so then
I think generally we do try, so that we can get through as many callers as possible. We do try to ask callers to limit yourselves to one question. So I would ask that you maybe pick your next top question, and then we'll move on to the next caller.
Thank you very much. I appreciate it. Like I said, I appreciate your patience. And this is my last question. Is the agency still utilizing enforcement discretion for promotional and sales of EU products up to 15 days prior to the submission of the EUA? Or could we preview our intention to our customers more than 15 days in advance? For example, coming soon, so that our customers know we're working on this important project? Or how far in advance can we do that?
So the policy that we have in place is regarding, excuse me, does indicate that you can notify FDA once you have completed validation and you are intending to begin to offer your test. So once you have notified FDA, you under that policy, you would then begin offering your test, and within 15 days, and this is for a molecular test, within 15 days you would submit your EUA request. For a serology test, it would be within 10 business days.
So it is the 15 days prior to submission. Am I understanding that correctly?
Once you have completed validation and notified FDA, we expect to see an EUA request within 10 or 15 days, depending on the time, the type of test.
Okay, so promotional materials, can we promote it prior to the 15 days?
The test should not be offered for use prior to completing validation and notifying FDA.
Okay, great. Thank you. And thank you all for your patience for my multiple questions.
Thank you.
I would like to remind participants that to ask a question, please press star 1 and record your name clearly when prompted. To withdraw your question, you may press star 2. Our next question comes from Lee Chow. Your line is open.
Thank you. Yeah, just a follow-up with the earlier question. I just want to clarify. And so for the standalone device, such as the approved home collection kit, the saliva, as well as the swabs. So if I already have a in-house EUA assay or LDT assay, what I need to do if I want to do home collection, I can just buy those standalone kit and that's it. There's no any additional test whatsoever, right?
So we have indicated that we do expect tests for home collection to be authorized prior to being used for home collection. So we would expect you to submit an EUA request for that. Excuse me, if you are looking to use one of the authorized home collection kits, which I believe the standalone home collection kits are only for nasal swabs at this point. I believe that we have collection devices authorized for saliva, but not home collection kits. And so if you do want to use one of the authorized home collection kits, the first step would be to reach out to that company to partner with them, and then they would be able to give you a right of reference to the data that you'll need to submit to FDA for your EUA request.
Oh, okay. So yeah, I just want to clarify. So as far as I know, there's a one home collection, the swab, the nasal swab kit that's been approved, right? Standalone kit. So then there's a saliva extraction solution. They got the EUA, the standalone as well, or maybe I got it wrong. So even
there are several standalone home collection kits that have been authorized for nasal swabs.
Okay.
There are no
for
home
saliva
collection kits authorized for saliva. There are, I believe, 3 EUAs for saliva collection devices, but they can only be used for home collection when they are part of an authorized home collection kit. They are not a home collection kit on their own.
Got it. So for the standalone, those swab, those kits, standalone, if I use those standalone ways together with my kit, with my test, do I still need to submit some EUA for FDA, or I'm okay?
We have indicated that we expect to see EUAs at this stage for all tests, all tests that are intended for use with home collection specimens.
Even with the standalone device, you still need to see the EUA application for that, right?
That's correct. And if there's a specific situation and a specific collection kit that you would like to get input on, you can send an email to the email address, and we can clarify.
Okay, great. Thank you very much.
Sure.
Our next question comes from Justin Binding. Your line is open.
Hi, can you hear me?
Yes.
Okay. My name's Justin Binding. I come from a smaller medical center, and we're seeing about using our current PCR instrument in these kits. We're currently working with the manufacturer that has an assay that is EUA, that has the EUA approval, and the software has EUA approval, but it doesn't have the emergency use authorization for the software with that instrument. We're working with them to see if about adapting it and developing a lab-developed test. But I saw on the FDA site that you're currently seeing about only approving emergency use authorizations for like at-home kits and collection kits and other ones that would be more widely distributed. Are we still able to receive the emergency use authorization, or would we have to go through CLIA and our accrediting agency for a lab-developed test?
We have indicated that we are not, that we are declining to review EUA requests for LDTs at this time. But what I would encourage you to do if you're working with the manufacturer of the the test kit is I would recommend that you work with them to possibly collaborate with them on the validation and have them submit a supplemental EUA request to add the components that you're interested in using to their EUA, as that would also help other labs that they want to use the same combination of components with that test kit. And then the EUA holder could update their authorization.
Okay, so that could still see approval if the manufacturer actually submits it just to amend their current EUA?
That's correct.
Okay, all right, thank you so much.
Sure.
Our next question comes from Lindsay Miles. Your line is open.
Hi, I have a question about manufacturing the viral transport medium. We are following all CDC guidelines for the sterility checks A and B. But my question is, do we actually have to perform, or is there a requirement that we validate or verify that sterility check? Or if we're following CDC guidelines, are we covered? If we are required to validate, how do we approach that?
You know, I have not looked at that guidance in a little while. I believe that for notification under the policy that we've put out, that we would just expect you to confirm that you are following the protocol and have validated it, and that you have internally validated that the VTM is sterile.
And so my question, though, is what does that mean? Because the CDC guidelines don't have a method validation process. So we are completely following their guidelines, but, you know, we're just wanting to make sure that we have everything covered, but there's not really any guidelines for a method validation for the sterility check that we are following per their guidelines.
Sure. Let me just see if I can find this very quickly. Otherwise, I would ask you to send a question in so that we can make sure that we are able to get you the right information there.
Okay, thank you. I appreciate it.
Yeah, I'm not seeing this in my very quick review of the information, and I don't want to take too much time on the call. So if you could send that into the mailbox, we can get you an answer to that.
Okay. And I'm sorry, one quick follow-up question to that. So I know you mentioned that it's just to validate that the VTM is actually sterile. So kind of how I interpret that is that the sterility check is the validation that the VTM is a sterile medium.
That's correct.
Great. And I'll follow up with sending to the mailbox, but that's how we're interpreting it, but we just want to make sure that we're following all guidelines and protocols that you guys have.
Great. And Chris, did you want to weigh in at all on this one?
Yeah, just briefly. You know, sterility is a statistical evaluation, right? So it's just a matter of how long and how much of the VTM you test. So I think that's perhaps why we're not having a kind of a concrete recommendation in that notification guidance, because I think there's multiple ways that it could be done. And so, as Toby mentioned, I think, you know, if you have a protocol in mind, I think we'd be glad to comment on it. We have done that in the past, but just keep in mind, I think there are, you know, multiple ways to take a look at sterility.
Okay. We'll send in the question and a follow-up to the mailbox. Thank you.
Great. Thank you. Thanks, Chris.
Our next question comes from Juliana. Your line is open.
Hi, Dr. Lowe. My name is Joanna Chen, and I'm with Hologic. Thank you so much for taking my call. We do have a question that we would like your help to clarify. We're starting to see many individual states publish travel requirements for travelers entering their states, and then these requirements are written in a way that they need to be a SARS-CoV-2 PCR test. Our test is one of the few non-PCR nucleic acid-based amplified tests, so it's one of the NATs, and this poses a challenge for the travelers, as they would still need to quarantine with a negative result from our test. And we've been seeing similar use of the term PCR used in places for descriptors for like amplified technologies instead of NAT or molecular IVD being used in like many programs for return to work and return to school at a local level. So we are hoping FDA could help us with two things. One being that if you could help us update the FAQs to recommend to policy developers that they could use generic descriptors such as NAT or molecular IVD when possible. So these tests are not solely based on PCR technology, and that we will not get the other non-PCR tests will not get excluded, and then that will help to educate the public health and policy officials at the state and then also the local level about the distinctions in between those two types of tests. And then another one, sorry, being that if we could have FDA look at the letter to clinical labs and HCP about antigen tests, if FDA could help clarify that NAT or molecular tests may be used to test antigen negatives instead of just PCR, or RT-PCR as recommended, that would be really great for us. Thank you.
Thank you for that. That's an important clarification, and I know that we do try to use molecular as a more general term typically, but absolutely we do refer to PCR tests at times when molecular may be more appropriate. If you have specific recommendations where you see information that we've put out and you think that we should change some of our wording, you can definitely send that in. If you can send that in to the mailbox in writing with your recommended edits, that is incredibly helpful for us, and we will definitely take a look at that.
Thank you so much. My leadership actually already follow up with you, and then we'll definitely send another email to the template address as well. Thank you so much, and we really appreciate your help. Thanks.
Our next question comes from Bruce Emanuel. Your line is open.
Hi, my name is Prince Emmanuel, and I have a very simple question. Do you have approved any EUA for the CRISPR-based this COVID testing?
I'm sorry, I'm having a little trouble hearing you. Can you repeat your question?
Yeah, sorry. My name is Prince Emmanuel, and I have a simple question. Do you have approved any EUA for CRISPR-based testing?
Yes, I believe that there are tests authorized with that technology, and yes, I believe it looks like there are two. And you can actually search on our on the EUA page by the attributes of the test, and CRISPR is listed for two of them.
So by any chance, I will be able to find also how long it took for them to get the approval?
We only list the authorization dates, not the dates that the EUA requests were received by FDA.
Thank you. I appreciate it.
Sure.
I show no further questions in queue.